All right, here is a quick review of the Hardy Weinberg equations and how to use them. So this coincides with Lab 7, Exercise 1, Part 1. So just a quick review, you can calculate allele frequencies using P and Q. So P represents the dominant allele, so capital T, in your population total, and Q represents the frequency of your little t allele or your recessive allele. And we know that if everything is in Hardy Weinberg equilibrium, P plus Q is going to equal to 1. So both their frequencies, so if one's 50% and the other one's 50%, that's going to equal to 1. For the genotype frequencies, essentially to get this equation, what they did is they put parentheses around this, and they squared both sides. And that's how you get the equation for calculating genotype frequencies. So as you can see down here, P squared is the frequency of homozygous dominant genotype, so big T, big T. For little t, we're getting Q squared, which you can see right here. And then the frequency for a heterozygous genotype is 2PQ. Okay, so if we know the number of homozygous recessives in a population total, we can actually solve for all of the other genotype frequencies in a population. So here is an example. So in our sample class data, we had 15 tasters. These genotypes could either be big T or big T little t. Both of them, whether they're heterozygous or homozygous, will be tasters because tasting is the dominant allele. Recessive, we know that all of them are non-tasters, which means that they have little t for their genotype. So we have 15 tasters and 7 non-tasters. That adds up to 22. So now we can solve for Q squared, so we can also get the genotype frequencies for the recessive, the heterozygote, and the homozygote dominant. So to solve for Q squared, we put the number of non-tasters over the total population number. So that's going to be 7 divided by 22, which equals 0.31818. So we can go ahead and add that into our table, and we know that the genotype frequency for that is about 31.8%. And if we multiply that by the total number of students, we can get the actual number of students that have that genotype, which is roughly about 7. When we multiply it out, we get 6.9999, so we can go ahead and round that up. Okay, next we have calculating the allele frequency for Q, because we already know what Q is, or Q squared is, to find P squared, which is the big T frequency. So since we know what Q squared is, we can just do the square root of that to get Q. So the value of Q, which is the allele frequency, if you remember from over here in this equation, so the allele frequency up here. So to do that, we just solve by square rooting the value of Q squared. So that 0.3818, that's right here. So when we do that, we get 0.564074. Okay, so since we know the Hardy Weinberg equation for allele frequencies is P plus Q equals 1, then we can solve for P because we already know what Q is and we already have 1. So all we have to do is subtract our value for Q from 1 and we get P. So P, the allele frequency for the dominant big T, is 0.4359. Okay, but to get the genotype frequency, we have to square it, because if you remember from our Hardy Weinberg genotype frequency, P squared is equal to the frequency of the dominant genotype. So we're going to go ahead and square 0.4359, and that equals 0.1900881. So we can go ahead and actually add that to our table. So that is our genotype frequency, so about roughly 20% of our population of 22. And if we multiply it by our total population, we get roughly 4.187, which we can just round down to about 4. So 4 students of the 22 have homozygous dominant values for tasting. That is their genotype. Okay, so lastly, we can use the values for P and Q, so our allele frequencies, if you remember, to find the heterozygote genotype frequency. So we know, once again, from our genotype frequency Hardy Weinberg equation, the heterozygote value or frequency genotype is equal to 2PQ. So what we can do, since we know the values of P and Q from up here, so the value of Q and the value of P, not squared, neither of them are squared, we can plug that in to 2 times P times Q, and we put those together and we get 0.497. So that's our last genotype frequency we had to calculate, and with that we plug it in, and then if we multiply that by 22 we get 10.8174. We can round up and we get about 11. So when we add that up to make sure our math is correct, we do get a total of 22 students, which is what we started with, and this adds up to, I think, 0.999 something, which is roughly 1, which is exactly what the Hardy Weinberg equation should say it should equal up to.